Okay, so now we are going to begin lipid metabolism, and this lecture will be on midterm 1. So this is a very short lecture just talking about the digestion and transport of lipids. We're also going to give just a very brief overview about what lipid metabolisms we are going to be touching on throughout this course. So with respect to lipid metabolism, we're going to talk about the production of our fats. So when we have excess fuels, your liver is going to take some of those sugar molecules and build fat from it. And then those, of course, will be stored for future use. We are also going to talk about the different glycerophospholipids and sphingolipids, not only how they're made, but actually what their functions are and their importance. We'll talk about our eicosanoids, which are going to be important in inflammatory responses. So our prostaglandins, leukotrienes, thromboxans, for example, are all lumped into this eicosanoid, and their precursor is arachidonic acid, which is an essential fatty acid. And then we'll talk about how cholesterol is made, the formation and breakdown of biosalts, as well as cholesterol's importance in making those steroid hormones. We do have a list here of fat-soluble vitamins, vitamin A, D, E, and K. And so these, again, have to be emulsified in some way in order to move through your blood. So they're going to be packaged, unlike your water-soluble vitamins that can just simply be released into your bloodstream. So our basic fat molecule is our triacylglycerol. And so the glycerol is the actual backbone shown here. And you have 3 carbons that make up the glycerol molecule, and on each of those carbons you will have a fat. So here is a fatty acid 1, we have a fatty acid 2, and a fatty acid 3. And you can see that they have different numbers associated with them. Here we have a 14, a 16, and this one over here is just, again, can be much larger. So we have a much larger fatty acid in that position too. And so the fatty acid attached to the glycerol is a triacylglycerol. And then, of course, for cholesterol, we have a sterile nucleus. And this is the structure of that nucleus. So all cholesterol products will contain this nucleus, whether it's biosalts, whether it's going to be a hormone, or just simply cholesterol. This is the nucleus of cholesterol. And then it'll have different things attached to it to form biosalts or hormones, etc. So as an overview, fatty acids, of course, we eat in our diet. And as everybody is aware of, our liver can make fats if there is excess sugars present. Fats can also be oxidized for energy. They are a major source of energy for the liver and muscle cells during fasting. They can be stored in adipose tissue, and those are in the form of those triacylglycerols that we just saw. They can be used to make our membranes. Our membranes need fat, and that includes our cell membranes, our nuclear membranes, our lysosome, etc. They all need fat. And so we can also make some biologic lipids. We can have hormones that have a base backbone of cholesterol. We have our eicosanoids, which are biologically active lipids. So fats do a lot of things in our bodies. Cholesterol metabolism, we're going to talk about how cholesterol is made. It starts with acetyl-CoA. And then cholesterol is going to be incorporated in membranes. That goes back to Biochem 1. It allows for fluidity in the membrane. It will also be a building block for other molecules that are important, such as biosalts and steroid hormones. Folate, shown here, is a biosalt. And so you can see that it has that steroid nucleus right here shown. And then it has a further carbon attached to that steroid nucleus, and that becomes folate, which is one of the biosalts that can form. All right, another important part of lipid metabolism and what we're going to talk about today is the digestion and transport of lipids. So again, lipids are not water soluble, so they have to be packaged in order to move through your blood. And lipids are going to be packaged into what we call lipoproteins. And lipoprotein is simply just a particle that transports lipids and bloods. And so what that means, what you should think about then with respect to lipoproteins is they have to have the ability to interact with water, because that's what your blood is made out of. And so they're going to have hydrophobic and hydrophilic regions. The hydrophobic regions are going to be sort of pointed to the core of the lipoprotein particles, and the hydrophilic regions obviously will be at the outer edges where they're interacting with water. So TAGs, or triacylglycerol, is also a major component in our diet. So when we eat fat, we're essentially eating triacylglycerol. And so this has to be digested, and it's going to be digested into free fatty acids and to monoacylglycerol. And then those free fatty acids and the monoacylglycerols are going to be absorbed by your epithelial cells in your gut lumen, and then they're going to reconvert them back into those triacylglycerols. The triacylglycerols then get packaged as chylomicrons, and in the lumen, remember, they are in something called a micelle. Micelles are emulsions that occur in the lumen with biosalts, and they are what transport fats in your gut, in the lumen of your gut. But micelles never get absorbed. Rather, it releases your free fatty acids into monoacylglycerols, which we'll talk about. And then, so this you can think of as part one. Part two of your dietary fat story is a package into a chylomicron. Chylomicron will have on their surface apolipoprotein B, and this is going to help to identify the different types of lipoproteins. Different types of lipoproteins have different types of ApoB molecules on their surface. This will be secreted into the blood. So your chylomicron gets into the blood, micelles stay in the lumen, chylomicrons get secreted in the blood, and then they're going to be matured. They are going to be matured by your high-density lipoproteins. Again, we'll talk about this. You have also your very low-density lipoproteins, VLDLs, and these are produced in the liver. So remember, chylomicrons are only made when you are actually actively digesting fats. They are not made in any other condition. Very low-density lipoproteins are going to be produced at the liver as needed. When there's too much glucose around, regardless of fed, fasting, if there's just too much, the liver can produce fats and package them into very low-density lipoproteins. Another important molecule with respect to the metabolism of fats is lipoprotein lipase. Lipoprotein lipase is an enzyme, and this is going to degrade the lipoproteins into free fatty acids. Lipoprotein lipase is never associated with the actual lipoprotein particles. What I mean there is very low density lipoproteins do not have lipoprotein lipase. High density lipoproteins do not have lipoprotein lipase. Lipoprotein lipase is associated with blood vessels that surround certain tissues that need to absorb your free fatty acids, namely your adipose tissue. So capillaries in your adipose tissue, for example, will have lipoprotein lipase embedded in their in the capillaries, and when these very low density lipoproteins come in there, the lipoprotein lipase can work on the triacylglycerols and break them down to free fatty acids. The other particle that's important is a high-density lipoprotein. So our high-density lipoproteins, or good cholesterol. So this is referred to as good cholesterol. And then your VLDLs are bad cholesterol. And so it makes sense. If you have a lot of very low-density lipoproteins, it means you're making a lot of fat because you have too much energy. High-density lipoproteins are not made when there's excess sugars. Instead, high-density lipoproteins are going to be important for transporting excess cholesterol back to the liver. And high-density lipoproteins have certain enzymes in them which allow them to exchange proteins and lipids with chylomicrons and very low-density lipoproteins. And this is important because your very low-density lipoproteins have receptors on them to interact with cells such as adipose tissue in the liver. But we'll get to all that. All right, so now just as a review of our tags, just keep in mind that tags, triacylglycerols, are going to be 3 fatty acids, and they're esterified to a glycerol backbone. Okay. In the fed state, you are going to eat your tags. So you eat, and then your tags are going to be digested to 2 monoacylglycerols and free fatty acids, and these will be absorbed into your intestinal cells. Your intestinal cells will generate new triacylglycerols, package them into your chylomicrons, and this will be associated with different proteins, your ApoB proteins, and that will be released into the blood. The liver will form very low density lipoproteins, and this is by making lipogenesis, making fat from sugar. Very low density lipoproteins are going to donate proteins to chylomicrons, and then they're going to bind to lipoprotein lipase on cells. Typically, this is going to be in adipose tissue. And also fat muscle cells have these, so they can break down triacylglycerols and use the free fatty acids. During the fasting stage, your fatty acids, glycerol, are going to be released from adipose tissue. Glycerol is a backbone for gluconeogenesis, and fatty acid is used to make ketone bodies or for energy. All right, so we're going to start this journey by talking about digestion and transport of your lipids. So we're going to talk about digestion of triacylglycerols. We're going to bring in bile salts, micelles, and then we're going to explain how those triacylglycerols get broken down, absorbed, then rebuilt, packaged, and sent out into the blood. We're also going to talk about the importance of the high-density lipoproteins in the process of digestion. This is not the process of cholesterol movement. We'll talk about that later, but this is in the process of actual digestion. So again, dietary triacylglycerols, or your tags, are a part of our diet, so you eat them. And then we're going to have different lipases that are going to digest these tags to generate 2 monoacylglycerols and free fatty acids. We have in our mouth a linguinol lipase, and in our stomach we have gastric lipase. And then as these fats move through our digestive system, they are going to emulsify with biosalts. And these are derived from cholesterol, so the backbone for these biosalts are cholesterol. So again, we have cholate, which is shown here, which is a biosalt, and that has that cholesterol nuclear backbone. So your lipid digestion is going to begin in the mouth, and again, that's going to be with that linguinol lipases, and it's going to continue in the stomach, where you have your gastric lipase. And then most of the digestion, however, is going to occur in the intestine, in which you have what's called chyme. It's going to be mixing with your lipases and your pancreatic enzymes and juices, and it creates that chyme. And so you have a collection of fats that form these particles, which will become micelles. Now, important for your small intestine are the release from the pancreas of lipase and colipase, and we'll talk about those. Okay, so lipids, of course, are globular. They are very nonpolar. They don't like water. They like to hide from water. And so biosalts are going to be important in breaking down those fats into small droplets, and this is called emulsification. And what this is basically doing, it's similar to having branching points of your glycogen stores. You're increasing the surface area, and so the enzymes can work at multiple points. And so lipases can interact more with these small droplets rather than this huge fat globule mass. Biosalts are going to be synthesized by your liver. They get secreted in the bile, and then this mixes with chyme. Chyme is gastric juices, food particles from the stomach, and of course your pancreas will also neutralize the gastric juices, and so that the pH of your small intestine is such that the enzymes in that area can work. So biosalts are going to be derived from cholesterol. Cholesterol is definitely hydrophobic, but when it's made into these biosalts, they're going to contain some polar aspects, and these polar groups are going to be hydrophilic. So biosalts is an amphipathic molecule, has both polar and nonpolar regions. And so you can see that your biosalts are going to interact with water as well as have the ability to interact with your fats. So when your stomach is going to empty its contents in the small intestine, when this happens, you are sort of changing the pH in the small intestine, and so an enzyme called cholecystokinin is released. Now, cholecystokinin is important because it stimulates the secretion of those pancreas enzymes and also contraction of the gallbladder. And so the gallbladder then is stimulated to release biosalts into the intestinal lumen. Your pancreas is going to release those lipase into the lumen. Also other enzymes, but we're focusing right now on fats. And then you start to get emulsification of your fats. Now, the biosalts are going to, you can think of them as acting as a detergent. They're going to bind to the fats as they get broken down. And the breaking down is by peristaltic action of your intestine muscle. And then as the action of your intestine muscles breaks down the fats, your biosalts will emulsify with the fat, and this increases their surface area again. And then you can have enzymes from the pancreas start to break fats down into individual fatty acids. So cholecystokinin is going to stimulate the release of those pancreatic enzymes. Two that are very important for fats is pancreatic lipase and another protein called colipase. Both are required in order to break down fats. Cholecystokinin is made by epithelial cells in the gut and the vagus nerve, as well as the parasympathetic nerve fibers. Cholecystokinin is mediated digestion, and it inhibits gastric emptying and decreases gastric acid secretion. The release of cholecystokinin is by a monitor peptide that's released by pancreatic acinar cells, and also by cholecystokinin releasing protein, or the presence of fatty acids and amino acids in the chyme. So any of those three can trigger the release of cholecystokinin. Another hormone that shows up here is called secretin. This was the first hormone to be identified. This is going to regulate the pH of your small intestine. So it's going to inhibit parietal cells from secreting gastric acid. It stimulates your bicarbonate production from centroacinar cells. And these are the cells, and not only those cells, and also the intercalculated ducts will produce bicarbonate. And this, of course, is to neutralize the stomach acid. Hydrogen chloride from the stomach is going to stimulate the release of secretin. And secretin signals the liver, pancreas to secrete biotin. An overall goal of secretin is to raise the pH so that in your small intestine, your digestive enzymes will be able to function. Okay, so our biosalts will bind to our pancreatic lipase and will actually inhibit pancreatic lipase activity. And this is by preventing the enzyme access to the substrate. So you can think of your biosalts as having two arms and two hands. One hand it's holding your pancreatic lipase, and the other hand it is holding your fat, and is keeping them separated. Now, the importance of colipase is colipase binds to dietary fat and to lipase, and this relieves that tension of the biosalt inhibition and allows your fats to have access to the enzyme. And this process increases the activity of lipase. Without colipase, lipase breakdown of fat would be very slow, too slow, metabolically slow. So colipase is, again, kind of like a coenzyme, and it's bringing the two pieces of the puzzle together so that you can get breakdown of your triacylglycerols into a form that can be absorbed. Pancreatic lipase is going to hydrolyze fatty acid of all chain lengths. It doesn't care. It can be 18, it can be 21, doesn't matter. It produces the free fatty acids and 2 monoacylglycerol. So you have a triglycerol, a diglycerol, and then finally you get on that second carbon a monoacylglycerol. Your cells can absorb a monoacylglycerol, and so it keeps the glycerol intact with at least one fatty acid. So enzymes that degrade these fats are called esterases. The pancreas will release the esterases that will remove the fatty acids from compounds such as cholesterol. Phospholipase A2 will digest phospholipids into a free fatty acid and a lysophospholipid. So here we have a cholesterol ester, and a cholesterol esterase is going to regenerate cholesterol. We have a phospholipid shown here, and we have a phosphate group here, and we have a phospholipase A2, which is going to release one of those fatty acids. So with respect to absorption, your gut epithelial cells can absorb fatty acids, free fatty acids. It can also absorb that 2-monoacylglycerol. And so what happens is you have micelles that form, which will be composed of those two forms of fatty acids that can be absorbed, as well as cholesterol and other lipids. And micelles, you can think of them as tiny droplets, and again, they get emulsified by bile salts. They are only found in the lumen of the intestine, and they form when the bile salts are in correct concentrations. The critical micelle concentration is 5 to 15 millimolar of bile salts, and so that's the concentration needed in order to properly emulsify your fats. And basically all this is showing you is you have your 2-monoacylglycerols, you have your biosalts, you get emulsification in your micelle. Your micelle will release your fatty acids that will get picked up. Your micelle will release your 2-monoacylglycerols. They will come together and reform your triacylglycerols. They will get packaged into what we call nascent chylomicrons. Nascent just means newly formed. And these nascent chylomicrons will have ApoB associated with them. They'll have other phospholipids, and of course your triacylglycerols. Your bile salts get released into the lumen, and most are going to be reabsorbed. All right, short and medium chain fatty acids don't require bile salts. Similar to short and medium chain didn't really require much to get oxidized by your liver. They're going to enter the portal blood directly and travel to the liver, bound to serum albumin, and then the liver will pick them up and use them for energy. Your chylomicrons are going to have your resynthesized triacylglycerols, and remember, it's those triacylglycerols are being formed inside of your epithelial cells from your gut. You're going to have activation of your fatty acids to fatty acid CoA in the smooth ER, and then you're going to have different apoproteins bind, as well as other lipids are going to bind, and then your chylomicrons get secreted into the lymph and blood. So you basically have activation of your fatty acids, you have fatty acid, and you have your ATP, which activates that, and then you have the CoA, so you get a fatty CoA. Triacylglycerol synthesis is going to start with that absorbed monoacylglycerol, and then you have your free fatty acid that was activated, and that's going to be added to your monoacylglycerol, so now you have a diacylglycerol. And then you have another fatty acid that gets added, and then you have your triacylglycerol shown here. Triacylglycerol with apoprotein B48 is going to be packaged into a nascent chylomicron. B48 is from the intestine. And so if you take someone's blood and you do a test for B48, you can in essence know how much chylomicrons are in the blood at any time. And of course you would only see chylomicrons after the fed state because they're only made after you digest fats. All right, so just a little bit about apoprotein Bs. They are important because they're able to differentiate different particles. So ApoB 100 is going to be in the liver, so it's going to be found on very low density lipoproteins as well as any remnant of that. So very low density lipoproteins will eventually be broken down into different size particles, and ApoB 100 will be on each of those particles. ApoB 48 is going to be from the intestinal cells, And this, again, is only going to be found on chylomicrons and usually only in the fed state. So they both come from the same ApoB gene. They just have different gene transcriptions depending on if you're in the liver or if you're in the intestine. So in the liver, you have a very long protein product, so that's why it has 100. And in the intestine, you have it truncated because you have a stop codon inserted. And so you get a shorter version of the proteins. But again, this can be just used to differentiate between the two particles. Now, nascent chylomicron again means that it is new. And so the nascent chylomicron is only from dietary tags, triacylglycerols. In the gut epithelial cell, in the smooth ER, you're going to get formation of your chylomicron. And then you're going to add to that apoprotein B48. Now, within this chylomicron, you can see that a majority of the chylomicron is going to be made up of triacylglycerols. You're going to have some proteins, and that's where that ApoB48 comes in. You're going to have cholesterol esters. You're going to have cholesterol. You're going to have different phospholipases, different other proteins. And that basically makes up your chylomicron. But what's key is your chylomicron is mostly going to be made up of triacylglycerols. Okay, so your particles are going to be transported in the blood, and you have to have additional protein, sorry, that got kind of hidden, to form mature chylomicrons. So your nascent chylomicrons will be released from your gut epithelial cells, and then your HDL particles will give them more molecules, and this will mature them. So the HDL particles are going to give them ApoC2. ApoC2 is important because it allows the chylomicron to interact with lipoprotein lipase, which is going to be on the surface of your cells in your adipose tissue and in your muscle cells, for example. And then also HDL particles are going to transfer ApoE. And ApoE is important because it's going to allow the chylomicron to interact with the liver. So the liver can pick up remnants of these particles and recycle them, digest them, get them out of the blood in essence. And so here's what it's just showing your high density lipoprotein particle has ApoC2 and ApoE. You have your ApoB48, your nascent chylomicron. Your HDL is going to transfer that ApoC and ApoE to the chylomicron, and this nascent now becomes a mature chylomicron. So what's the fate of chylomicrons? So again, they're going to be matured by the high-density lipoprotein particles. They get digested at the capillary walls, and this is typically going to be in adipose tissue. So during the fed state, you're in a storage stage, and so your fats are going to go to storage. So lipoprotein lipase in the capillary cell wall, remember it's binding to that C2 that was donated by your high-density lipoprotein, is going to digest your tags into free fatty acids. So the free fatty acids will bind to serum albumin in the blood, and then the fatty acids can go into the muscle for energy, or it can go in the adipose tissue to be stored. And the remnants are going to go to the liver through ApoE and be digested by your liver cells. So here we have our intestine epithelial cell. It's going to release that chylomicron. The chylomicron is going to go into the blood. The chylomicron has been activated or matured by your HDLs. So we can do an HDL maturation. And so by that C2, which was given to it by your HDL particle, it's being able to interact with lipoprotein lipase. And so it releases the free fatty acids that can be picked up. Muscle will use for energy. Adipose tissue will store. You can also release your fatty acids, and that can be picked up by your liver. Now the chylomicron remnants, sorry about that, are going to have ApoE, and then this is going to allow it to bind to the receptors on your liver cells and be removed from the blood. So in essence, here is the function of these lipoproteins. Your chylomicron is going to be transporting dietary fat. Your very low density lipoproteins are going to be produced from the liver, and mainly from dietary carbohydrates, so sugars. So when you have a high carbohydrate diet, you're going to produce fat. Your intermediate density lipoproteins are remnants of VLDL, so they'll have that ApoB100 on their surface. Your low density lipoproteins are remnants of intermediate density lipoproteins. Again, they're going to have that ApoB100 on their surface. So this has 48. This will have 100, 100, 100. The low-density lipoproteins are important because they're able to move around a bit more, and they have a very high concentration of cholesterol. So they're going to be important for your brain and tissues that need to make cholesterol-laden hormones, for example. So they'll be able to donate to areas like that. High-density lipoproteins are going to be produced in the liver as well as in the intestine, and they're going to be able to exchange different proteins and lipids with others, and they also help to return cholesterol back to the liver. Your chylomicrons are going to be produced in the intestinal cells, very low density lipoproteins from the liver, and then IDLs, LDLs are going to be remnants of your very low density lipoproteins. Now, just as a side note, olestra is an artificial fat substitute. It tastes like fat, but it's not metabolized. It's a sucrose with fatty acyl groups that are esterified, and so it's going to be resistant to hydrolysis by lipases. And so it basically just passes through you. And the side effect is, however, that it carries lipid-soluble vitamins with it. So if you do eat a lot of this, I believe it's the yellow sugar package, you have to make sure that you get enough vitamins because it will take vitamins with it. Okay, so that is the end of this lecture, and we will continue our lipid story in the next lecture.